So hopefully you've had a chance to think about homeostasis a little bit since we last met. An interesting thing to think about is if we have these feedback systems in place that are responsive to disruptions, we used blood pressure as an example, how is it ever possible that an individual develops high blood pressure? It's an interesting thing to think about. If we have a deviation outside of our ideal range, it should be corrected back towards the ideal. And in most cases, that happens. Depending on the size of the deviation, again, the compensation is proportional, but if the deviation is too large, it can cause disorder or disease. So in this example, not in this example, I guess I deleted that animation. Imagine the temperature straying too far towards the top of the graph, or blood pressure rising too much instead of 120/80, which you've heard of anecdotally before, and I won't talk about the details. Maybe you've registered 140/90, 145/90. That's class 1, class 2 hypertensive. Well, negative feedback loops should reinstate the norm. But if, and this is the catch, if you continually insult that system with the same type of disruption, eventually the body gives up. It says, I'm trying to regulate around this set point, and 23 hours out of the day I'm living up here. I'm going to move the set point up. So there is some plasticity in that negative feedback loop response, and that's how we can develop hypertension, high blood pressure. The continual insult, however it happens, to increase pressure inside the vasculature raises the set point, and now we regulate about that 130/85 instead of 120/80. I know why it didn't work. I'm on the wrong slide. That's why. There we go. That's what I just described. The body will eventually change the set point to regulate accordingly. These are in the slides on Moodle. You can feel free to copy them down if you want to. So if we continually change, if we continually cause a disruption, that does affect our overall health. The set point changing is now not ideal on average. Perhaps it's ideal for our situation because there's no other out, there's no other option, but it's higher than the average organism would like. Let's leave that discussion there. And I'm stalling for a few seconds to allow you to write everything down. I still see heads bobbing up and down. We'll leave our discussion of homeostasis there. We have an understanding of the two major types of feedback loops, how they differ, and how they're similar, receptors, processor, effectors. And we're moving to a discussion on types of tissues and the organization of tissues in the body. This is going to be helpful for underpinning our discussion of the integumentary system, which is a big focus of Lab 1 coming up next week. Also keep in mind, microscope practice still on. It was on yesterday, on today, until 5, is that right? Ish?
Still about 40 that haven't picked up a lab.
40 haven't picked up a lab manual. Not the ones that are in lab on Monday, I imagine. You have that and you've read through it already. You've made notes that you're going to review over the weekend in preparation for your Monday lab. I'm sure of it. If you haven't picked up your lab manual, maybe you have lab next Thursday. You've got some time. Please do that ASAP. See Matt in 422 in JBV. So tissue types and organization. Tissue types and organization. So tissues, like we talked about, are made of cells and the stuff between cells. The cells themselves are complex, and it's the net sum of cells and fluid between cells and proteins and other paraphernalia that make a tissue. And the tissue overall will have some function. And we'll leave it there. We won't go to any higher order level discussions yet. We'll discuss how some cells are connected briefly. And while all these characteristics are the same, the resulting tissue is different, wildly different, depending on where you look. Bone, very dense, hard, durable. Adipose tissue is semi-solid, stores energy. Blood is a tissue, which is fluid. It has no real structure. So wide range of tissue types that are made from the different and the constituent cells and other factors that make up that tissue. So we think of a tissue as a grouping of cells and other stuff. Whatever the tissue is, it typically has a common origin and then a common function with the other cells in that tissue. Usually these groups of cells are of a majority. There's a majority type of cell within a tissue, but they don't all have to be the same type of cell. You have multiple types of cells within a given tissue. This will become more clear as we explore today. There are 4 basic types of cells in the body. And there's variations of each type of cell, and the combinations in varying amounts of those types of cells make for different tissues that give it different functions, but there's only 4 major types, which is great. Less to understand and memorize at the foundational level. 4 basic types, but multiple arrangements and variations of those 4 basic types. Those 4 basic types are epithelial, connective, muscular, and nervous cells. You'll examine the majority of these in Lab 1. Not muscular, but the other 3. I think muscular comes later.
You'll see that in the integument.
We do have muscular cells. Fantastic. You'll explore all of these cells in Lab 1. So epithelial, in very general terms, is a type of cell that lines surfaces or compartments, cavities, ducts, spaces within the body. Epithelial cells usually create a tissue that is a border between 2 environments. Internal environments or external environments. Connective tissue, various types, various functions. You'll see many of them in Lab 1, which is good because we're not spending a lot of time talking about them today. So you'll have to review some of the textbook material and certainly the lab manual preamble before you go. We'll spend a lot of time talking about epithelial cells, and I'll leave muscular and nervous until we get to those respective sections, which are coming up fairly soon. But connective tissue can be cartilage, bone, fascia that connects and wraps bones and muscle within the body, adipose tissue. Muscle is any cell that acts to generate force, that contracts, that changes shape and moves in response to some stimuli. And the stimuli is usually from a nervous cell, nerve tissue. This orchestrates and communicates between the brain and different parts of the body via what we call action potentials. action potentials. And I'm introducing that term now. We won't discuss it. Maybe you know that term already. We'll talk about it at length in the nervous section, which is in a few weeks' time. But action potentials is, it's the name for the electrical signal conveyed by a nerve cell. It's the message. So these are our 4 main tissue types. And there's variations in each, but these are the classes. Now I want to talk quickly about cell junctions, because the combination of cells within a tissue, I think, needs this discussion in order for you to grasp the context of how cells are arranged. This and the next slide were not in the slide set on Moodle, but I'll put an updated version online for you. It doesn't really matter, it's not in depth. I just want to show you the types and arrangements of different connections between cells. So cell junctions, here we have listed 5 main cell junctions, and this is just a schematic of an empty cell. The flaps are the plasma membrane, so the container of the cell. And we have some finger-like projections at the top that we don't care about yet, and a purple thing at the bottom that we don't care about yet. What we care about are the connections between these cells. So look at wherever the membranes touch each other or something else. First type of connection is what we call a tight junction. Tight junction. And you might still be able to see it under the shading. It's a weave-like pattern. There are proteins that span through the membrane on each side, through both membranes, and weave the membrane space closed. It seals the space between the 2 cells. It's like shrink wrap or heat sealing on plastic packaging. It makes sure that nothing can get through, nothing can be transmitted between the cells through that space. It seals that space really nicely. Second type of connection is what we call an adherence junction. These are circular bands that usually wrap all the way around a cell. And the proteins here are thicker. The bands are what we would call a plaque. Aggregations of proteins that stick out across the membrane, and they anchor with complementary proteins on neighboring cells. And they fix the cells in place. They anchor them in relation to neighboring cells. It's almost like the seal on a Ziploc bag that closes the 2 sides so it doesn't open. I really like these desmosomes. These are little spot welds. They don't seem to do anything at first. And we're not going to see what they do probably for a long time. Maybe not ever. You might talk about these more in cell bio. These are little spot welds that anchor the plasma membrane at one specific point. 2 neighboring cells anchored at one specific point across their plasma membranes. And it's an anchor point for the cytoskeleton, so the scaffolding of the cell. And so from this desmosome, these little filaments sticking into the cell on each side, there will be long strands and long filaments extending from this connection point to other desmosomes at different points within the cell, making a really intricate lattice work that crosses the cell, provides stability. What's really cool is it allows the movement of the cellular machinery. The cytoskeleton is a location for movement of organelles and substances and enzymes within the cell. It's a way things can move around. The fourth type is comparable. Hemidesmosomes, they're half a desmosome, or they look like half a desmosome. They achieve the same goal of anchoring the cell, but they don't connect to neighboring cells. They connect to the, well, I have it here, the basement membrane, which is something we'll discuss soon. You don't need to know what it is immediately right now. But they anchor the cells in place. Make sure that they don't change positions within a tissue. And lastly, gap junctions, which are connection points that are leaky. Gap junctions are a collection of small pores between two neighboring cells. And these small pores allow the cytoplasm to move back and forth. So the fluid within the cell, some of the ions within the cell, this will be important when we get to the nervous section. But larger molecules, glucose, things like that, can't slip through. Enzymes, proteins stay on their respective sides, but the fluid and ions can move through. So it allows communication between two neighboring cells, which is important when you're sending signals that need to execute a task. The action potential is an example of that signal. And we'll talk more about that later. So these are 5 different types of cells. We refer to a couple of them later on in class, but this is the level of our discussion. It's a higher order level. There's more detail in the book that you can go into if you'd like. So any questions on these before we move on? No? Okay. Let's do a quick comparison of epithelial and connective tissues before we dive into a discussion on epithelial tissues. Remember, connective tissues are largely the focus of lab. There's much information in the book. These are the 2 most widespread cells or tissue types in the body. There's a wide range of structures and functions. We'll get to the other tissue types, like I said, in their respective sections, the muscle cell and the nerve cell. So immediately, at face value, there are a few large-scale differences that you can observe. Major differences are immediately obvious. 1, the number of cells to the space around them. So this is an example of a histology slide, and usually the dark round circle in the middle is the nucleus of a cell. It's bordered by a faint, somewhat dark staining envelope, which is the membrane around the cell. And each of those cells, you can see on the left, is packed tightly together in epithelial tissue, whereas it's not in connective tissue on the right. There's a lot more space, a lot more stuff between those cells, which we call collectively the extracellular matrix. The extracellular matrix. Proteins, strands, the scaffolding, the things in the space between the cells. Less in epithelial, there's more of that in connective tissue. What's not immediately obvious is that the blood supply is very different between these 2 tissue types. Epithelial tissue has no blood supply. Connective tissue has lots of blood supply. And I imagine that's what these little structures are here, although I'm not exactly sure. I'm pretty sure these are blood vessels. So epithelial tissue not having blood supply could be a problem, except that in practice, these 2 types of tissues are intimately related. They are almost always adjacent to one another. And so the epithelial tissue has access to nutrients from the blood supply in the connective tissue. And that occurs by simple diffusion. It's a little tricky to recognize also, but the epithelial cells on the left here have a defined border, and that's meant to indicate that they are usually at the surface. They aren't contained by any other cell type or tissue type. They usually face into a duct or a tube, or out to the surface, or into a body cavity, and there isn't another tissue that overlays the epithelial cell, or the epithelial tissue. That's not the case for connective. So being that epithelial tissue is a lining tissue, a covering tissue, you probably have an idea of where it's located already and what organs might consist mainly of the epithelial tissue. We'll talk about general features and then get into that discussion as well. So general features of epithelial tissue. This slide shows you epithelial and connective tissues. It's not going to make sense when I tell you that it's ciliated, pseudostratified, columnar epithelium, but that will become clear by the time we're done this course, or the class. Hopefully also the course. General features of epithelial tissue are: 1, cells are often aligned in sheets. 1 sheet, 5 sheets, 30 to 50 sheets in some cases. Not usually that many, but cells are aligned in sheets and are closely packed together. They are tightly connected to neighboring cells by many different types of cell junctions and anchored to a membrane, the basement membrane that we saw on that cartoon schematic drawing. We've already identified that it doesn't have blood supply, it's avascular, but it does receive nerve supply. It does receive signals from the nervous system. It also is proliferative. Mitosis occurs frequently. New cells are constantly being produced to replace any cells that are damaged or that are lost. And so as a lining, the epithelial cells, the epithelial tissue, the most obvious role is at the skin. That's a pretty obvious lining in the human body. Epithelial tissue also usually is used or is present in glands. They have a role in secretion of different materials as well. And technically glands, the inside of glands is still a surface. Or they line blood vessels, line organs in the thoracic cavity like the heart and the lungs. Epithelial tissue is part of the serous membranes we talked about last class. Many different roles. They can all generally be categorized as lining surfaces or in forming glands. The organization is depicted here. General organization between the epithelial cells and the connective tissue. Notice the apical surface, the free surface, the surface pointing into a cavity or outside of the body. Here it's at the top. There's a lateral surface of these cells. These are Also columnar cells, they're shaped like columns. We'll talk more about that on the next slide. Lateral surfaces point towards neighboring cells, are the sites of connection. And then the basal surface at the bottom connects to the basement membrane. If there are more layers, if it's not just one layer, then the apical or free surface is the top of the most superficial layer, the uppermost layer, and the basement membrane, or the basal surface, is the bottom of the deepest layer of cells. So you can see the nerve supply, the lack of blood supply, but the close association with blood vessels in the connective tissue. Any exchange of gases, nutrients, waste products occurs between these two regions via simple diffusion across the basement membrane, which anchors the epithelial cells in place. The basement membrane has two types, or two variations, two divisions, if you will. The basal lamina is actually secreted by the epithelial tissues, or the epithelial cells. It's a mixture of collagen, fibrous tissue, laminin, sticky proteins that help keep the cells in place. They provide anchor points for those hemidesmosomes. And then the reticular lamina is similarly secreted by connective tissues and largely provides a border, a barrier to movement of large molecules. Proteins, enzymes, glucose, things like that. Ions, gases, and waste products can move freely. Large molecules cannot. So this is the orientation that you will commonly see in its simplest form. As a lining tissue, the form can become more complex. The types of epithelial tissues that we'll observe are characterized by arrangement of cells and type of cells, as shown here. Major type is the lining epithelium, which is what we're looking at here. There's also some glandular epithelium. I'll show you a few quick examples of that coming up, but it's not the majority. So arrangement progresses from simple on the left to more complex, stratified on the right. Simple allows for easy diffusion and absorption of nutrients, of gases. Pseudo-stratified in the middle are actually a version of simple. There is really only one layer, even though these cells are extended and deformed somewhat. And what makes it pseudo-stratified is that not all of the cells extend all the way to the apical surface, to the free surface of the epithelium. Some cells stop halfway, so it looks like there are layers, multiple layers of tissue, when it's just that some cells don't extend the entire way through. Stratified, many layers of individual cells. The more layers, the thicker the tissue, and the greater the protection against abrasion, UV damage, temperature, etc. So think of the skin and where heavily stratified or more layers of stratified epithelium might be. Now the types of epithelial cells differ as well. They're not all just round cells or square cells, as indicated here. We have squamous epithelium, which are very flat and thin cells that are really useful in allowing quick diffusion or movement across them. They're very thin, there's little obstruction to the movement of elements across these cells. Rapid passage of substances. Cuboidal cells are shown in the middle. They look like small cubes, and columnar cells, extended, stretched versions. These are very similar to cuboidal cells, but being that their internal volume is larger, they have a greater capacity for absorption and secretion of whatever they happen to be absorbing or secreting. So you will see things like squamous epithelial cells in the lungs to allow for rapid exchange of gases. Makes sense? Thin cells, quick passage and diffusion. And columnar cells in the digestive tract. Large secretions in the stomach that prevent the cells from being degraded by stomach acid, or specialized cells in the small intestine that release digestive enzymes. And then various configurations of the apical surface that help it do its job. Again, get to that later. I want to show just quickly, if I highlight for you the idea that I proposed on the last slide, if there are multiple layers, the apical surface is this top free layer, basal surface at the bottom. The ones in the middle do not. They have an apical or a basal facing surface, but they're not technically those surfaces. So there are many excellent examples of each combination in the book. Table 4.1 gives you a nice rundown of all the different combinations, their general functions, the locations, and what they might look like. I would encourage you to review that before Lab 1. And if we go back to the slide that I showed you originally, it might be a bit more clear now to imagine the pseudostratified columnar epithelium with connective tissue and a basement membrane. We can see connective tissues here and here, and the epithelial layer at the top. Pseudostratified because the nuclei are not all at one level. They're not clustered towards the bottom. It looks like they're spread out in multiple layers. If this were columnar epithelium, the nuclei would all seem to line up near the basement membrane. This is tissue that's characteristic of the upper respiratory tract. So ciliated pseudostratified columnar epithelium allows for protection. The cilia are little finger-like projections that wave around and prevent dust particles from entering the lungs or damaging the epithelium or getting lodged in the small airways. They move in unison to get those particles either to be expelled by coughing or sneezing or what have you, or to the back of the throat to be swallowed and disposed of in other means. You'll see many more examples of these in lab, and a nice cheat sheet to help you with the naming combinations is shown here. What I didn't mention was transitional cells. Transitional cells can be columnar, they can be cuboidal, and they can change back and forth. An example are cells in the bladder that change shape as the bladder expands and contracts. They might appear to be cuboidal in one instance or columnar in another. Notice no pseudostratified squamous or cuboidal tissue in cells. A nice cheat sheet to help you. Now I briefly want to mention the glandular epithelium. This is an artist rendition of a sweat gland, which would still be lined by epithelial cells. And in brief, a gland is a pocket, an invagination. It's a duct with a secretory portion of epithelium that's specifically designed to secrete and expel something. In the case of sweat, that is to secrete and expel sweat. Of course. Glands are not only sweat glands. Sweat glands we think of at the skin. Sweat is released onto the skin. They almost seem visible. There are endocrine glands that we'll talk about hopefully near the end of this semester that release hormones directly into the blood. And that's the difference. Endocrine glands release whatever their secretory compound is to the bloodstream directly, whereas exocrine glands, of which a sweat gland is one type, release it to the surface of something. Here, the surface of the skin. There are tables here. I'm not spending a lot of time on this. There are many different kinds of glands by whether they branch or not, and the shape of the duct, and the shape affects how the secretions occur. That's not really a major focus. What I want to highlight is that epithelial cells can form this other type of structure, and that the structure has a specialized purpose. So here we have an example of an endocrine gland in the thyroid. In the thyroid gland, simple cuboidal cells line the gland, and they're secreted into the neighboring blood vessels. There's some precursor that's stored, that is taken up and absorbed by these cells, processed, and then the active hormone is expelled into the bloodstream. That's different than an exocrine gland. As shown here, it's actually quite difficult to see the sweat gland itself. It's this very small area on the right-hand side of the cube. There's a squiggly portion at the bottom, which we call the secretory coil. And then this long, thin piece that extends all the way up to the surface of the skin, which is the sweat gland duct. An example of an exocrine sweat gland. As one contained unit, something is produced within the coil, and as more of it is produced, the excretion is pushed upwards towards the surface. That's how you sweat. Create very salty primary fluid in the coil, and as you produce more and more, it pushes that fluid up until eventually you release sweat as you know it on the surface of the skin to be evaporated to help regulate body temperature. Notice the epithelium, here not simple, but stratified. And the actual process in making primary fluid and making sweat, you don't need to really know right now. It's kind of neat, but I might be biased, since most of my research revolves around sweat. Any questions on these glands, epithelial cells, characteristics in general? No, it's review. Good. You all know this well and could teach the class if you were up here. Fantastic. Okay. We're going to start briefly an introduction to the integumentary system, because some of you will not have any more lecture material before you get there to your lab on Monday. A lot of what you do in lab will then have to come from the preamble to the lab, in the lab manual, which is fantastic and comprehensive. So the integumentary system, largely made up of epithelial and connective tissues, and is a barrier to the external environment. It contains fluid, proteins, cells, tissues, organs, everything within the body, and protects it from things in the external environment. Wear, tear, pressure, heat, UV light, you name it. Trauma, infection, disease. Who, as a tangent, who's been to the Body World exhibit? Anybody? couple people. Does anyone not know what Body Worlds is? A lot of people don't know what Body Worlds is. So it's not common. It's not. I think it was here last year. Was it actually here last year? It was in Halifax last year. Not Integumentary. We're not a big draw for exhibitions or concerts or anything. Body Worlds is. I can show you what it is on the next slide. I just wanted to make sure that you were prepared for what I was going to show you on the next slide. Body Worlds is a fantastic exhibition of human anatomy. It's plastination of cadavers in various forms, orientations, and all the structures within the body that are sectioned and presented in a way that you never get to see them in real life or even in many academic labs. It was at the Royal Ontario Museum when I was back in Ontario. I got to see it there. I incidentally saw it while I was taking undergrad anatomy, and so I walked in thinking I knew everything and was so skeptical of these exhibits. I was wrong. My criticisms were unfounded. But it shows you, if you're sensitive, you might want to just take a minute. It shows you things like this. This is a Body Worlds exhibit that differentiates or separates the integumentary system from the other systems in this one person. And I'll have a number of different examples of what you can see at the Body Worlds exhibit. I did look, and the exhibition is not coming back, which is unfortunate. I thought we could organize like a trip or something. It'd be kind of neat. Mostly it's in Germany right now, so if you happen to go to Germany over Reading Week, you can check it out over Christmas. It doesn't do anything to really help us understand the integumentary system, other than the fact that it covers about 2 square meters, weighs about 10 to 12 pounds in an average adult. It can be 1 to 2 millimeters thick on average across the body, up to 4 millimeters thick in some places. You don't get that information from the skin suit this guy is holding. It contains all of the structures necessary to support the integument, so epithelial, connective tissue, and the accessory structures, sweat glands, hair follicles, nails, any number of structures. And the main focus, like we mentioned, is protection, containment, and also largely to produce vitamin D for use by other tissues in the body. So the skeletal system, for instance, needs vitamin D to properly sequester calcium. Vitamin D comes from the integumentary system. When we happen to be out in the sun. It's also the site where we receive sensory information about our surroundings, provides pleasure sensations or pain sensations, heat sensations, most sensations that we need to interact with our immediate surroundings. So structure of the skin is generally broken down into 2 main levels. The epidermis and the dermis. And we might have time to get through the epidermis today, but maybe not even that. 2 main structures, although there is an accessory third structure that we often discuss at the same time, but that is not part of the integumentary system. The epidermis is the most superficial layer made of epithelial tissue. The dermis is the layer immediately deep to that layer, made of connective tissue, in the orientation that we described previously. And as you would expect, the epidermis has no blood supply, it's avascular, whereas the dermis has a large blood supply, it is very vascularized. Deep cuts bleed. Small scrapes don't often draw blood if they only scratch the epidermis. Deep cuts that delve into the dermis will probably also hit the vasculature and release blood. That third layer is the hypodermis, or the subcutaneous layer. I'll often abbreviate it, and I think it's abbreviated in the book as sub-Q. It is deep to the dermis and largely adipose and areolar connective tissue. So adipose, we have an idea what that is. Stores energy, stores fat, can be quite large in some cases, can expand and grow, can shrink. And areolar tissue, you'll see good examples of this in lab, largely the packing material of the body. Provides strength, elasticity, and support to a wide variety of tissues. And then below the subcutaneous tissue is usually the fascia, the tissue covering the muscles and the bones. It doesn't do it justice to describe it in words, so you can see it here. Notice the epidermis, the most superficial layer, organized, usually stratified epithelial cells. The dermis with its heavy vasculature and nervous input, immediately deep to the epidermis. And then the subcutaneous layer, the yellowy, globule, fatty region at the bottom. And there are a number of really good examples in the book that show real images of the dermis and the hypodermis and the subcutaneous tissue. A really cool electron micrograph as well, which shows the small structures as they would appear if you zoomed in close enough. Let's not go further. We'll start discussing the four main types of cells in the epidermis and what their characteristics are to give the skin the characteristics that you know skin to be like. We'll talk about those when we get back to class on Tuesday. Otherwise, have a wonderful weekend and read your lab.